Hi, my name is Victor Alvarado, and my project is the comparative analysis of Zamia leaf anatomy and vulnerability to herbivory damage. Now, these are going to be the agenda of the things that I'm going to talk about today. Now, for the Zamias. The Zamias are a subgenus of the Psychodials order. They are a group of gymnosperm organisms, which means that they grow their seeds outside. They're not protected by anything like a fruit or an ovary, and they have existed for over 300 million years. In the Zamia genera, about 86 species up to date has been identified and studied to a certain degree. Like any other plant, these cycads interact with their environment in many ways through pollination, resource competition, nutrient cycling, herbivory, and etc. And some of these interactions are influenced by unique characteristics of each plant, such as the size, the number of stomata that they have, how the leaves may look, how they may grow, fortunately to the sun, now, for the conservation of these cycads and why I may have chosen them. Now, these cycads are very slow and unpredictably growing plants, which means that many species are vulnerable to endangerment if they are not already threatened. And I pulled this information from the IUCN Red List of Threatened Species. The integrifolias are native to Florida, Bahamas, Cayman Islands, Cuba. The Vasquezes and Perforatia are both native to Veracruz in Mexico, and the decumbens are native to Belize. All of these are Caribbean regions. Now, this is the Humus the butterfly. It is the most prevalent herbivore, if not the only one, of these zamias. Now, the Zamia tigrifolia is specifically the host plant of this butterfly. Now, these adult talas, the females, will seek out flushing cycads, which means cycads that are currently growing new leaves to lay their eggs. Now, some pest measurements were taken as a last resort due to the environmental and ethical consequences, semiochemicals, which means it would be using a combination of the host plant volatiles and sex pheromones of the butterflies to trap the males and disrupt the mating process. And there's another one, which is introducing beneficial predators. Although this didn't work out because studies have shown that this specific butterfly has psychocin, which is a toxin found within the cycads that the larvae eat, that they are able to eat and contain it inside their body throughout their lifespan. Now, these Atala populations would regularly fluctuate whenever the cycads would grow new leaves, which is typically only during the summer months. But in Montgomery Botanical Center, many different species of zamias are grown, protected, and studied all in a contained area. Because all of these different species come from different parts of the world, that means they all grow new leaves at different times of the year, allowing a food source to be continuously available for the larvae. Some species are threatened by larvae feeding highly, such as the Integrifolium and Vasquezi, and others are almost completely ignored, the Cumbens and Perforatia. I included some pictures here and with the source from where I got them, as along with the graph, that these photos were taken in the same area 5 years apart. As you can see, the damage is so prevalent. Now, the research question is, how does the epidermal anatomy of these Caribbean Zamia species affect herbivory by the larvae? And the objectives of this project are to perform a comprehensive comparison of these four species and identify any possible trends or similarities in the epidermal layer of young and mature leaflets using SEM techniques. Now, these were some of the materials and methods that I used. In total, 24 samples were collected from Montgomery Botanical Center of these leaves. 8 initial samples were collected on September 15, and they were stored in an oven kept at 43 degrees Celsius for 5 days. Then they were removed, stored in silica gel, and transported to the analytical electron microscopy lab in FIU MMC. Now here shows the machines that were used for the initial sample preparation. This is the sputter coater because the samples have to be sputter coated with gold for 10 to 12 minutes. This is to make the samples conductive for the SEM imaging. Now, this is the second round of sample preparation where the 16 rest of the additional samples were collected on October 4th. They were similarly stirred in silica gel and taken to the lab. But for this round, chemical fixatives, dehydration sequences, and critical point drying techniques were found. 1/4 to 2/3 inch cuttings were made from the young and mature leaflets. They were fixed in 25 milliliters of 99.9% methanol for 10 minutes to fix the cell structure, followed by two 30-minute dehydration sequences in anhydrous ethanol. And this is a critical point drying machine that was used, as long as this is the brand of the machine. We use 99.9% methanol as the transitioner fluid and liquid CO2, which was stored in this tank over here, as the bleeder. Now, these are results of the young adaxial side of the Zamia leaflets. As you can see here, I've labeled PC, ST, and I have what they mean down here. For pavement cells, ST for stomata, SC for subsidiary cells around the stomatas, and T for the unicellular non-glandular trichomes, which are found. As you can see, the biggest differentiation between all of these is the furfuracea. As you will see in further slides, this is the adaxial face of the furfuracea of the young leaflet, and it will look the same on the abaxial face. The trichome density is just so much higher compared to everyone else's. And one more interesting thing that you will find is that in none of the other adaxial faces of these leaflets were stomatas found, because in the Zamia species, stomatas are rarely found on the top side of the leaf. It's mainly on the bottom side. But as you can see here, hidden behind trichomes, you will see subsidiary cells surrounding the guard cells and the stomatas of the leaf, which is something interesting to be noted. Now, this is the adult adaxial leaflets. As you can see, they're pretty uniform in nature. They all look very similar to each other, with the only one differing from the rest of them, the Vasquezes. They have this undulate pattern of pavement cell structures. Now, this is the abaxial side of the Zamia leaflets, of the young Zamia leaflets. As you can see here, in contrast to the adaxial stomata found on the integrifolias, the abaxial stomatas follow a similar pattern. They follow the same rows, just one per row. Now, the most interesting images were when I did the imaging for the adult abaxial faces, the bottom sides of the adult leaves. The most drastic change between young and adult leaflets were the Furfuracea, as you can see, the almost near complete loss of trichomes. The Vasquezes have the most puzzle-like pattern of pavement cell structures, and integrifolias and decumbens share same patterns of stomatal bands and the parallel venation going down. Now, to the discussion part. What made these anatomical features describable of the plants? The stomatas on both sides of the young integrifolia leaflet can be linked to increased nitrogen levels from endosymbionts or cyanobacteria, which are found in the roots of integrifolia plants. And these endosymbionts help fix nitrogen for the plant. Now, these increased nitrogen levels, there has been studies linking them to increased rates of photosynthetic activity, which could possibly explain why a plant would want to maximize its tomato quantity in young leaves. For the decumbens and Vasquezi, their unique pavement cell structure can signify a higher need for water retention and protection of the specialized cells under. Now, the one young leaflet that seemed to be the only one that has adapted a defense mechanism against pests or herbivores were the Furfuracea, which their high trichome density could have been a defensive adaptation against pests. Now, what is the importance of the anatomical features on these leaves for the pavement cells? These parenchymal cell barriers basically provide protection between the atmosphere and the specialized cells below. The guard cells found around the stomata help control the opening and closing of the pores to facilitate gas exchange and transpiration. Trichomes provide an additional layer of protection against abiotic and biotic hazards. What this means is it can help protect against the UV rays of the sun, and for biotic hazards, it can help against pests, as they basically help to form a hard layer between the actual leaf layer and the pest. These subsidiary cells found around the stomatas help increase function and protection of the guard cells, which they're also known as accessory cells. The stomata intake carbon dioxide and release oxygen. Now, the connection to herbivory interactions is that the stomatal quantity and the act of them closing and opening the stomatas can affect the leaf temperature, water, and nutrient content, which can affect how viable of a food source a specific leaf may be. And the trichomes, like I said earlier, act as a chemical barrier because in other studies, trichomes have been known to also exude chemicals as a defense mechanism when it is being consumed. Now, some future directions I would like to take this project in is I would like to do mass spectrometry and liquid chromatography for this, which what this basically means is that I would like to do an analysis of the carotenoid, which are basically a type of pigment cells, identification of the ketocarotenoids, and I would tie that into the Lepidopteran evolution of red wavelengths plus vision, as these specific type of lichenid butterflies have been seen to have evolved to be able to see red long wavelengths and short wavelengths. I would like to test different environmental conditions effects on the leaf anatomy of these plants, such as exposing them to different sound concentrations of salt, different intensities of light, and different concentrations of nutrients. For my acknowledgements, these are my references that I use for this project. Thank you.